So today on Nature's Always Right, we're going to be talking about a soil test on my field, giving you guys the nitty-gritty information with my good friend Brian Maisenbacer, earth grower on Instagram, who is a professional soil scientist and consultant and a farmer as well. He's just a great guy. I met him at the Natural Farming Conference with Chris Trump, and that's one of my favorite things about his services as a soil consultant, because he can give you the recommendations for natural farming inputs after looking at your soil test. So this episode is going to be about how do you do a soil test, what are you looking at when you see it, and why you might want to hire a professional to help you look at that to then put out the proper inputs onto your field. It's going to be really cool to see all of his recommendations. He did a full write-up. There's just like so many things that we can learn from a soil test, but we're also going to tell you about natural ways that you can determine the health of your field. You don't have to use any of the microbiology or any of this modern science to determine many things about your soil. So let's get Brian in here and talk more about it.
Yeah, thanks for having me. I'm really happy to be here and to help you take a look at these soil tests.
So I got my test from Logan Labs. I know lots of farmers, you recommend Logan Labs because they do a great job, and then the price is pretty good too for what you get.
Absolutely. I really like Logan. So when we're looking at a soil test, we're looking for a few different things. One of the things that's important to me is I can get someone on the telephone. Logan, their customer service is really great. The price is good. So when we're looking at price, we don't want the most expensive. We also don't want the cheapest test that we can get. So they're a good kind of middle of the road test. And they have quick turnaround time, and they're also using some of the most advanced equipment for doing the testing. So they're up to date on everything, quick turnaround time, right price, and customer service is important. So that's some of the reasons I like working with them.
Excellent. So there's two types of soil reports that you may want to get. Just so you got your standard soil report, and then you have a saturated paste reports. Could you tell us kind of the difference and why you might want to get both or one of them?
So one of the first reports that Logan does, this is kind of their standard soil test. This is called a malic 3 testing. So they use certain types of acid to extract the nutrients from the soil and then run that through a very advanced meter that will produce these numbers, these percentages. So our malic 3 test is very much like our battery, right? So what is there, what's available? Well, not necessarily what's available, it's what's there, everything that's there. So calcium, magnesium, potassium, our trace minerals, our phosphorus, and also our estimated nitrogen release. So the first thing that I'm looking at when I'm looking at one of these tests, we start at the very top here. I'm looking at our total exchange capacity. So this tells us a lot. This total exchange capacity is telling us the amount of nutrients that are held within the cation exchange capacity of the soil. So it tells us our electrical charge of the soil. It tells us how much nutrients can be held there. So when we see a number like 5, anything below 10 is easily leached soil. So it doesn't hold on to nutrients very well. It doesn't create that chemical bond that happens between cations and anions. So when
So is that happening like the clay molecule or the sand molecule is there, and then say there's minerals surrounding that molecule of soil? Is this is describing the electrical connection between that?
Yeah, so a clay molecule has multiple sites where the other nutrients can attach to that. So this is telling us about the ability for our soil to hold on to those nutrients on those attachment sites. So when we're below 10, that's saying a lot about the physical structure of the soil. Like, because it's below 10, this is likely very sandy soil, right? And it's letting go of those nutrients. It lacks the clay molecules for it to bind. It also is lacking in organic matter. So one of the next things I'm looking at is organic matter. A glazed over pH, we'll come back to that. But the organic matter here is below 4. So for really great agricultural soils, we're looking for 4 plus for organic matter.
I'm trying to go to 10. 10's my number.
If we can be up there, that would be amazing. But at minimum, we're kind of looking for a 4. So the organic matter tells us a lot as well. So when I'm looking at organic matter, I'm considering this total exchange capacity. And because when we can raise our organic matter, we can raise our total exchange capacity. But also when we raise organic matter, when you come down here to the bottom, estimated nitrogen release, we'll see that number go up as well, because a lot of our available nitrogen is stored in our organic matter. So these are some of the first things I'm looking at. In terms of pH, this soil is actually in a pretty good range. We're at 6.9, 7, 6.3. So when we make an average of those, we're kind of in the right range where majority of your nutrients, because of that pH, are in an available form. They're readily available and can uptake. So when I'm looking, I just go down the line here. The next thing I'm looking at is sulfur. Sulfur, generally, we're looking for a minimum of 25. So we're at 7. So that's a bit low, and that's going to cause nutrient lockout. It's going to cause other issues with your trace minerals and nutrient uptake. And low sulfur is very common around the whole country. So when they stopped using some of the nasty chemicals that they were using, though they're still using a lot of them, we used to have acid rains, sulfur rains in the United States, and those would actually add a lot of sulfur and where it had a benefit to agriculture in some regard. But since we stopped doing that, we're seeing really low sulfur numbers around the country. That's actually really common. Next thing I'm looking at is phosphorus. So there's different types of phosphorus tests depending on the part of the country you're in. Malic 3 is a common one. There's also the Bray test, there's Olson, and those would be, you'd use them for different reasons depending on your soil type. For this one, we went with the Malic 3, and usually we're looking for at least 250 pounds an acre. So you're in a pretty good range on this part of the field, this field 3. And this is all really one field. So when we make an average of it, this part of the field likely didn't get as much of the fertilizer and things that prior to when you came to the land, right?
I think it's an interesting little note, because field 3, where I took that, was at a high position, the highest part of my field. So there'd be more runoff there. The nutrients are running down. Field 1 and 2 are lower parts of my field. You know, we can make an educated guess that the cation exchange, it's not holding those nutrients, it's the water's draining it down, and then it's collecting down these lower areas. This is an example of a thing that you can kind of determine from a soil test just by looking at numbers like that.
Absolutely, that's a really great insight, that this was the higher point of the field, so things are leaching out. So in general, you're in a decent place in terms of phosphorus. So same thing, we're looking at calcium here is the next thing we're looking at. And you have a decent amount of calcium in terms of what's there, right? So because you have low exchange capacity and you actually kind of have high calcium, that's what we call calcareous soil, right? So there's a lot of calcium there in that parent material, in that sand and what's in your soil, but it's not necessarily calcium that's super available. It's not in a water-soluble form. Though there's good calcium there, which is holding an electrical charge, it's not necessarily calcium that's readily absorbed by the plant. Next thing we're looking at here is magnesium. So for magnesium, you're a little bit low. So we're looking at a ratio here of calcium, magnesium, right? So we'd like to see calcium in terms of base saturation between 60, 70%. And we'd like to see magnesium between 10 and 20. So this ratio helps with that electrical charge that helps plants grow. So when we're out of ratio, we'll start to see issues. And so because you're actually higher than we're aiming for calcium, that can cause some issues. And then we're lower than you're aiming for in terms of magnesium, that can cause some issues as well. And then looking at potassium here, you're low also. So potassium is easily leached. It moves around quickly. Then we're just going to go down the line here, and the next thing that I'm looking at is trace minerals.
Sorry, before we go on, could you explain this? It's the part that says exchangeable cations and base saturation percentage. So that section is kind of goes together and it's all related.
Absolutely. So the base saturation, this part in gray, is the percentage that will go into solution from this type of test, right? And it's not water soluble necessarily, but it's what will hold in solution. So base saturation and exchangeable cations, this is based on the Albrecht method. So when we're talking about interpreting soil test, there's different lenses we can look through, and one of them is the Albrecht method. And that's the method that I use. I do use other methods depending on the type of soil. Like I'd mentioned, different parts of the country will use different phosphorus tests, and you have to interpret that a little bit differently. So when we're looking at the rest of this test here, the next thing I'm looking at is trace minerals. And in general, around the whole country, boron is low. This is a common thing. Your iron is in a decent place. That's common. Your manganese is low, your low copper, your low zinc. Aluminum is in a fine place. Cobalt and molybdenum. So when we're talking about like purchasing a soil test, cobalt and molybdenum are add-ons generally to a soil test, and they're really important because cobalt and molybdenum, they work synergistically with soil bacteria to actually pull carbon from the atmosphere, to fix carbon. They're critical to the process of carbon fixation. So when you're planting cover crops of your legumes and all these kinds of things, though the plant does play its part in that, there is specific bacteria that helps to facilitate that, works with the plant, and they are using cobalt and molybdenum as well in this process. So this is go ahead.
Okay, so what that just told me is if I can increase my cobalt and molybdenum, then that will help me to increase my organic matter percentage more quickly. Is that correct?
Yes, that it absolutely could mean that. And cobalt and molybdenum are things that we need to be very careful with because they're heavy metals. So you can absolutely put too much. So we need to be very careful in the way that we put these things. So when I'm talking about natural farming, right, one of the main ways, and really one of the only ways that I like to add trace minerals from that perspective is using seawater, sea salt. A product like C90 is good, which is just fancy sea salt. So all of the trace minerals that we need, over 90 trace minerals, are in good quality sea salt. C90 is a kind of a cool product, and the way that they harvest that sea salt makes it a little bit of a unique product. But absolutely, we can harvest ocean water. We can use other types of sea salt, absolutely. And that's going to contain molybdenum, cobalt, these other trace minerals that we're lacking in an available form. So when we're talking about a salt, right? So a salt is a cation and an anion attached to each other. So when a cation and an anion attach to each other, that become positive and negative charge. That becomes an available nutrient. So when we think about electrolytes, right? So we consume electrolytes. Maybe you drink a Gatorade or something, right?
Hopefully
not. But
you can make your own from sea salt and lemon juice, and you can make your own.
Absolutely. But when we're talking about electrolytes, that's exactly what they are. They're salts. They're cations and anions attached to each other that carry an electrical charge that help us to hydrate, right? The same thing happens with plants. So that's why in the hydroponic world, a readily available nutrient, they're called salt nutrients, right? Because they're a cation and an anion attached to each other, and the plant can get it. So when that process is kind of cobbled up within the soil, and cations aren't attaching to anions properly, you're lacking in organic matter, you have a low CEC, you're leaching off nutrients, you're not getting nutrients in their available form. So, but when we add a layer to our perspective, this is just talking chemistry, right? When we add a layer to our perspective with biology, the biology has the ability to take nutrients like this calcium that's not necessarily super available and consume that and store that in their cellular structure and then make that more available for the plants. So when we're talking biology, sometimes people would like to say that these tests are irrelevant, but it's not necessarily true because it's showing us what the potential of what the biology could do. And so that's really critical when we're looking at these tests, is to take that biological perspective. Yeah, I'm high in this, but I know, and it's not necessarily an available form, but I know that if I add fungi can consume calcium stored in their cellular structure, be consumed by nematodes and protozoa, and then excreted, and then now it's something available and my plants can get it, right? So these are interesting perspectives when looking at this kind of test.
Yeah, and Albrecht, he's from the early 1900s?
He's from the early 1900s, and he's one of the formative figures in creating some of these soil sciences.
But he didn't know about biology, which is so interesting because that's what in the last 20, 30 years has been discovered with Elaine Ingham, and that's where all this knowledge that Brian just said is like the bacteria and fungi, they're the ones that unlock everything for us. And I think that's what's so interesting. I used to say, and to some extent I still believe, that soil tests are irrelevant or they're not necessary, but it's a tool that we can use. Technology is a way that we can accelerate growth or accelerate all these different things. And as you're seeing, we've only barely touched this soil test, and you can already see so many different things that we could do to this soil that we now have more information. So this gives us more information, more tools. So it is necessary, honestly, in a way, because now I can get my field to a way better position way faster. Could I just rotate my animals, feed them minerals, apply biology with natural farming? Yes, and that would be amazing too, and it will make these numbers better over time. But if we have a little bit more prescriptive thing going on, it'll happen more quickly. I just wanted to make that point. I wanted to make that point in this video that it's like there's many ways to achieve this, and it just like the time scale I think is one of the biggest variables that changes in there, and maybe cost.
Yeah, absolutely. And one thing to keep in mind is these soil tests, any test you get from the doctor, any type of testing, is not 100% accurate. So it's all about how you use this data and interpret it and come up with an action from it. So the more that and that's why it's important to not work with a ton of different labs, because all the different labs are going to give you slightly different numbers because they have slightly different protocols, slightly different equipment, and slightly different process. So when you can work with the same lab consistently, then you can begin to correlate the data and then come up with an actionable plan. And not all soil consultants are created equal. That many soil consultants maybe only take a biological perspective, maybe only take a chemical perspective, and often both are overlooking physical structure of the soil. So when we're talking about a full baseline approach, we're looking at chemical, biological, and the physical structure of the soil and how they push and pull on one another and how they can affect each other. And then from interpreting this, we can find what are these determining factors. What is the smallest thing I could do that will actually create huge change across the board? And these are the kind of things that I'm looking at. What is the most critical thing that's going to make the most meaningful change for the land?
That's a great point. And that's, I'm spending a little bit of money up front. I paid $150. I can't remember the exact price. $150 for these soil tests. But with this information I'm now getting, if I could make my pasture 20% more yield, That's 20% more meat that I can grow, which means more money for my farm. I'm eliminating even more weeds because as I change the biology of the soil profile, the nutrients available there, different seeds are going to germinate in response to that. If I graze my animals correctly, different plants are going to respond to that accordingly. And we'll get into it later, but we're also going to take a look at some of the weeds that are plants that I have in my field, and then how those plants correlate to nutrient deficiencies or nutrient excess. This is just like another tool we can use that doesn't even involve technology to take a look at our field and see what's going on.
So I'm going to go ahead and pull up another report here. So one thing that was one of the big teachings of Albrecht was that he believed that human health and soil health were connected. And this is something that is running true across the board. And the data is beginning to prove this. And this is something that he was talking about almost 100 years ago. So though the science has progressed since him, he held a perspective that's actually reigning true as the science progresses. So the next test that we're looking at here, this is a saturated paste test. So saturated paste is, there's a few different reasons you would use a saturated paste test. So when I'm looking at a soilless medium that doesn't necessarily have sand, silt, and clay, this is the test that I like to use for looking at nutrient content of like a potting mix or a soilless medium. But also this shows what is water soluble, what's readily available in terms of salts. So people will use a saturated paste test to see salt content as well, because salts are readily available nutrients, right? So when we're looking at this test here, some of the things are running true that when we're comparing them, sometimes this test will say something that's almost the opposite of what the other test is saying, right? Because the other test shows everything that's there, not necessarily what is readily available, right? So this test is showing what is water soluble, what's readily available nutrients that the plants can use right now, right? So when we're looking at this, pH is the same. The first thing I'm looking at is sulfur. So yeah, sulfur is low. We knew that looking at the other tests, and this is they're lining up, right? Same thing, phosphorus. Phosphorus is showed to be high on that other test, but it's actually, when we're looking at this, it's not necessarily in a super available form for the plants. Calcium, though calcium showed high on the other test, it's not necessarily in a form that the plants can use. But when we consider biology can take that stuff, process it, and make it available. So when we're adding biology, we know that we are mining some of that nutrition that's not necessarily water soluble. They will consume it and put it into their cellular structure, be consumed by other organisms, and then create nutrient cycling, right? This is really an important thing to consider when looking at chemistry tests, because when we can adjust biology, we all of a sudden open this door of all of these things that are available for us that wouldn't necessarily be if we were lacking in biology.
Right. And if I was to just add more calcium out on my field, unless it's in a form that's immediately available, like a liquid calcium or something, it just would have no effect. If I put gypsum out in my field, which all these old school farmers will tell you to do, lime your field. Never listen to them. Never put that on your field. Maybe not never. I'm always extreme with things, but you would rarely want to do that.
Any good ecologist, any good consultant, we like saying it depends, and so Elaine Ingham would tell you the same thing. It depends, but she would tell you absolutely no in terms of using this stuff, because she subscribes to a biological perspective, right? So my perspective is a little different. So depending on what the physical structure of that soil is, I may consider a calcium supplement, because something like gypsum can be quite helpful in breaking up compaction and balancing a calcium magnesium ratio. So if you're dealing with soil that's very high in magnesium, which you're not,
And I don't have clay either.
don't have much clay either, adding something like gypsum could help to, because gypsum has calcium carbonate, but it also has sulfur in it, and sulfur helps to leach down magnesium. So if you're very high in magnesium, when you're high in magnesium, your soil is really tight. It's tight soil, and that's the soil old school farmers will say, like, oh, high magnesium soil sticks to the bottom of your boots, right? That's is an old kind of adage because it's tight soil, and something like gypsum has the ability to help to balance that ratio and break up that compaction, but that's not necessarily something you would use everywhere, right? And something like lime is usually used for balancing pH, which is something that we don't put a huge amount of focus on in terms of living soil, because
biology is adjusting.
The biology will adjust it. So when we're looking at this saturated paste test, like I was saying, these numbers don't always line up with what we're seeing on the other tests. So it's a completely different perspective of what's everything that's there and what's water soluble, right? So that's creates a big difference in our perspective of what we're looking at. And then we add this biological component, it changes it even more, right? So this is a perspective that's a little bit unique. I mean, there's other consultants that take this kind of perspective when looking at these tests, but it's not necessarily commonplace. Just looking at this test, we're low sulfur, we're low phosphorus, low calcium, low magnesium, even though it says high magnesium on the other test, right? Low potassium. It says high phosphorus on the other one. So that's what's there, but not necessarily what is readily available. So
Yeah, this is the argument for why you probably should get a saturated paste test with all of your tests. By doing both tests, it makes the overall test more accurate because you have more things to look at in combination.
absolutely. And then being able to interpret and understand why they're different and how they affect each other, and then it starts to kind of make sense, right? Oh, that makes sense. And they start tying together.
Right, like that's why I was able to figure out certain things as he was talking, because I have some base level knowledge. I know the basics, but this is where the expert comes in, that he can fill in all these hundreds of holes that I'm missing in my knowledge gap, because this is what he does day in and day out.
It takes a lot of practice and looking at a lot of tests to be able to begin to be able to interpret them in a very meaningful way to make actionable change. Because a lot of the time when I first started looking at these tests, I mean, it didn't make a whole lot of sense. It takes a lot of time to really chew on what these numbers actually mean in a situation, and then be able to come up with an actionable plan based on that. This can be quite challenging.
Absolutely.
So that's the importance of the saturated paste test. So I recommend getting both. So if you're working with Logan, you can get the complete plus extras. So the complete plus extras will include a saturated paste test. It's going to include a Malic 3. You can ask for different phosphorus tests depending on the part of the country you're in. And also it includes cobalt and molybdenum, and also selenium and other trace minerals.
Other clues. I'm just looking at all these things as little clues that give you a fuller picture of your soil profile.
Absolutely. Each one is a snapshot in time, and that tells us different things. So the more snapshots we can have, we can widen that window. But also, there's this kind of fine line where more data can be good, but the wrong kind of data can be confusing and is actually a form of waste or wasted energy looking at data that we can't actually do anything with, right? So having the right kind of data that's meaningful, that can create actions, is the kind of data that we should be looking at. So I'm going to go ahead and pull up another report here.
And then we're going to get into his recommendations after this?
We did 3 different microbial reports. So when we were looking at the saturated paste, the Malic 3 tests, there's 3 reports side by side. So field 1, field 2, field 3. We did the same thing with the microbial. So this is just one report, and we'll only look at one because they were all pretty similar in terms of biology. There were some differences in terms of chemistry because of the slope of the hill and some of those other points we talked about, but in terms of biology, there wasn't huge variance from across the field. This is one of my microbial reports. We use microscopy to generate these reports. So the main things that I'm looking for when I'm doing one of these reports, the first thing I'm looking for is fungi and also nematodes. So nematodes are quite large. You can see them almost immediately when you put them under the microscope. Same thing with beneficial fungi. You can see this stuff fairly quickly. So as we can see in Steven's field, because he hasn't been there very long and he's first just getting going on really all of his processes, there's not a huge amount of fungi there. Because this says 0, like I said, no soil test is 100% accurate. That doesn't mean that there's not fungi in his field. It means that I didn't see fungi in the sample that I took. But considering that, with the protocols that I use within a sample of really great soil, you should be able to see quite a bit of fungi in one drop of that solution. So this tells a lot about the soil, that there's not a huge amount of fungi there, because if there was, I would have seen some. But then there was a decent amount of bacteria. So depending on the client, if I don't see fungi, there's not a huge amount of need to actually start counting bacteria. Because without the fungi, I'm not going to be able to generate a fungal bacterial ratio. So all that we're looking at, and I counted them for you for the purpose of reviewing these reports, but these numbers don't mean a whole lot, because bacteria is always there no matter what. Bacteria is very opportunistic. It's going to show up regardless. But what's interesting when we're looking at bacterial numbers when we don't have fungi is if those numbers are very low, that's really interesting, because it's saying that there's not a huge amount of a food source for those plants, for those bacteria there. So you're in a range this is really common to be between 800 and 1,000, So when we're looking at these fields, I do 5 different fields, and you can do more. People do up to 20, but
And that means 5 slides.
so that's one slide, 5 different positions of,
In that slide.
and they're random. You pick random spots. So on 5 random fields, these are the counts of we're getting for bacteria, and these are really common counts. Like you can see way higher than this, but there is good bacteria there. And in terms of the type of bacteria, we don't really have the ability to fully identify any kind of bacteria with microscopy. We can use trace genomics, we can use DNA testing to begin to determine those things, and even that's not 100% accurate, right? So what we do is we look at the taxonomy of them, we look at the shape of them, right? So majority of the bacteria that I saw in these samples were small cocci, cocci. So small little round bacteria, very common bacteria.
Lower level stuff.
Yeah, and so other bacteria that we can look at are for like rods. Rods are more leaning on being bacillus or yeast, other types of bacteria. So the small cocci they're very common. They're the ones we see the most. Some of those likely are archaea, which are the oldest bacteria that we know of that make up a lot of life as we know it. But without doing DNA testing, we can't know what they are, and we don't really need to know. necessarily. Though that information is interesting and likely will reveal insights for us as the science progresses, it's not super critical in terms of nutrient cycling, right? So to go down the line here of this test, the things that are really important that I'm looking for are fungi, beneficial fungi. There's other types of fungi, so narrower fungi that have a narrower diameter, that are lighter in color, that don't necessarily have septa, so striations that will break that fungi apart into separate segments. Those fungi tend to be not the best fungi. The best fungi are wider in diameter. They have a little bit darker color. They may be tan or brown. They'll have those septa, those striations in between the segments of the fungi. So when we're looking for beneficial fungi, those are some of the things that we're looking for. And also we're looking for fungal spores. So when we're talking about looking at IMO, an IMO may not necessarily have, especially IMO3. IMO4 is a different story, but IMO3 will be mainly fungal spores. It looks like a sack of marbles, kind of, when you're looking at a grouping of fungal spores. So when I'm looking at soil, I'm looking for those as well. You see those a lot in compost. It's really common in compost. But then in terms of negative stuff, actinobacteria, when that starts to take over, that is anti-mycorrhizal, it's antifungal. So when we're seeing huge amounts of actinobacteria, that tells us a lot as well. There was little to none present in your sample. Actinobacteria can have benefit to certain types of plants. So brassica plants associate with actinobacteria and can find a synergistic relationship with actinobacteria. But in general, we're looking to have very low amounts of actinobacteria and have a little bit more beneficial fungi. So you're in a pasture setting. We're not looking for crazy amounts of fungi, but I'd like to see 1-to-1 would be great, or a nice balance of bacteria and fungi is going to be very helpful in a pasture setting. Then looking at omyces, these are kind of more pathogenic. They look kind of like fungi, but the science is showing that some of these things may not be as negative as we actually think they are. They serve other functions.
Right. And I think I would compare that to parasites, like in sheep, for instance, or parasites in our gut. Like we have some of these organisms that we would consider to be bad, but they do seem to play some sort of role in our immune systems, and they need to be there in some way. So it's like this whole microbiology world, it's so complex. We've barely scratched the surface, barely. And I think that, proceeding with some caution of just some humbleness that we don't understand jack about this world yet. We're just getting into it. But that's what's so interesting and fascinating about it too, is that it's so undiscovered and so new. And I just love how it interplays with natural farming, which would be like the complete opposite philosophy of like not drilling down to this nitty-gritty reductionist stuff, but there's still so much information there. And that's like the marriage of Western and Eastern sciences, I think, is like the match made in heaven. And that's like why I really appreciate the approach that you're taking. You're taking the Albrecht Western approach, you're taking the natural farming Eastern philosophy approach, and you're putting those together. And I think that's really where the best science and soil science is going to come from into the future as we progress.
And because of that, the type of microbial reports, that my perspective on them is a little different than, say, a soil food web consultant. Yeah. A soil food web consultant generally are trained to see anaerobic or facultative anaerobes as a negative thing, and in natural farming, that's not necessarily the case. And we're seeing that some of these anaerobes and facultative anaerobes, a facultative anaerobe can kind of bridge that gap. They can go aerobic, they can go anaerobic, and they can kind of wiggle in that zone.
so can fungi. There's facultative fungi too. And that's the thing about, we keep discovering more and more things. So to ever make a definitive statement that anaerobic bacteria is always negative in soil, to me, it never made sense to me. Especially as you would go deeper into the layers of soil, there's going to be less oxygen, but there's still microbes there. So how does that work? And then all of the fermentation of the foods that we eat and drink, it's all anaerobic. Our gut is anaerobic. So, and we know that our gut biology is related to this soil biology. So there's a lot of interplay there, and I think that because of the infinite complexity of nature, I think it's important to never make a super definitive statement, I guess.
Absolutely. We have to be dynamic, we need to be flexible, and we need to be ever expanding our understanding, because that's the way that nature works. It's dynamic. So when I'm looking at this report at the bottom here, though, flagellates, amoeba, ciliates, and nematodes. Those are important. We want to see those guys. So the flagellates and the amoeba, they are consuming bacteria and fungi, and they're excreting available nutrients. So these predators, so on the bottom of the report here, I said no predators, right? So poor nutrient cycling. Without these predators consuming the other smaller bacteria and the fungi, the bacteria and fungi are gathering mineral nutrition. And they do this from signals from the plants. So plants will signal them in the rhizosphere to go gather particular nutrition, put that into their cellular structure, and then they'll be consumed by the flagellates, the amoeba, and the nematodes that will excrete available nutrients. On top of that, the plant roots through the rhizophagy cycle actually will consume some of these themselves. The rhizophagy is a blossoming field of understanding within microbiology that the plant roots themselves actually eat bacteria and eat some of these different characters, which is very interesting. Whole topic in itself. But when we're looking at this report, the things that are concerning to me is lack of fungi, lack of predators. Bacteria, it's what it is. They're gonna be there, they're gonna come, they're gonna go. You'll see those fluctuate. If you're low in bacteria, add sugar. Watch it go right back up. You put a liquid IMO on, there's plenty of sugar in there. You're gonna see those bacterial numbers jump. You'll also see fungi jump when you do that. So there's this interesting thing that happens when you use something like liquid IMO. Of course, you're adding a fungal inoculum that's going to start to take hold in that soil, but you're also adding a sugar source. So you're going to see a spike in bacteria before you're going to see a spike in fungi, because that's kind of the balance of how it works. It's harder to establish fungi. And what's a really cool thing about IMO is when you look at IMO 3, yes, there's tons of fungal spores in there, but there's also a ton of lactobacillus. Lactobacillus is a yeast. So yeasts are kind of the building blocks. Yeast a lot of the time will show up before fungi does. So when we are adding that inoculum, we're adding not only a ton of fungal spores, but we're adding yeast, and we're adding the building blocks to create a functioning system. And usually what will happen is once you have yeast, once you have your different fungi present, your bacterial variety, the type of bacteria will start to increase. You'll see more diversity. And then once that is functioning, you'll see nematodes starting to show up. You'll see protozoa starting to show up, because now they have a conducive environment. They have something to eat, right? They have a place to function.
As you're speaking, something that I try to teach people is to look for the patterns in nature, to understand nature at a deeper level. And because of my knowledge of other parts of nature, I can apply those patterns to the micro, to the macro. Nature is an endless fractal, and these patterns apply everywhere. So he's telling me you have no predators. What does that mean out in the garden? If we have no predatory insects, no predatory animals, what is going to happen to my garden? Tons of bad guys are going to come in and take over. There's no one to stop them. So I imagine that if I was to plant plants in my field, if I try to do a garden there with not adding compost or anything like that, there'd probably be really high disease and pest pressure if I tried to grow in this soil. So just applying that pattern is so similar. And then what he just talked about with the yeast is preparing the way for these higher level organisms. That's going on in our gut, that goes on with like protein synthesis, hormone synthesis in our body. There's all sorts of places in our world where that's going on. Weeds are there to set up shop for a future plant. That's why we're getting the Bermuda and the crabgrass and all these things, that the bad guy grasses. In the future, as you improve your soil, those start to disappear. And he also told me to do a 1-to-1 fungal bacteria ratio. And that's kind of based on the idea of ecological succession, that the field or the prairie, the place where you're going to grow vegetables, is going to have more bacterial content. And then all of nature is trying to go towards the more established ecosystem, which would be trees like softwoods, then hardwoods, then you get your redwood forest, and the peak of ecological succession is 1,000-year-old forest. Okay, so all of nature is going through this cycle, and we're applying that knowledge to our soil test. So I know that in my field, the edge of a forest, and then into the pasture, having that 1-to-1 50-50 bacteria fungal ratio is kind of that ideal that when you test a really good healthy field, that you're going to find that there. So I just wanted to add in those couple of details, but that's what's so cool about nature, and that's why I never get sick of learning about this stuff. That's what, like, to me, that's like what proves God to me, is like, how are these patterns in everything? Like, it's so amazing. And anyways, yeah, let's keep going.
We're on the same page, man. And the idea of succession, right, is something that's really important to understand, and to even deepen that understanding a little more. We're talking about like the soil food web, right? So there's all of these different predators and prey that are in succession, and that's in the soil, but that also includes things above the soil. It includes birds, it includes all of the animals passing through your property, it includes your livestock, and beyond that, it includes you, right? And that is often a perspective that we glaze over. The fact that we are actually very intimately connected to this system, and that we're in every way are part of it. The way that these different patterns push and pull on each other, they affect us, they affect our health, our animals' health, and through the act of understanding and participating in them, not only will the farm become stronger, but we'll become stronger, right? Mm-hmm. And that's an understanding and a perspective that I take when I'm looking at all these reports, or when I'm helping farmers, is you're part of this, you affect this. So what are you able to do? How are you able to participate with this and maintain that healthy perspective, right? So the next thing I've got on the bottom here, so this is kind of a quantitative assessment. On the bottom here, it's more of a qualitative assessment. So, and you notice I have NA here for the FB ratio. It's because there was no fungi to be found. So there's no ratio to be had. So one of the other things that I'm looking at when I do the samples is I use colorimetrics to determine a percentage of humic acid content. So this is gauged from 1 to 6. So you're at about a 3. You're kind of midway in terms of humic color. I'd like to see that at a 5, 5 or 6, right? Really great compost 6, it becomes black instantly in solution.
You're talking about running water through this, right? And then the water that comes out.
Yeah, so when I do these samples, I'm adding water, so I'm making basically a compost extract, and I'm putting one drop of that onto a slide and looking at 5 fields. And when I do that, I'm shaking up my sample, I pull it up, and I use my colorimetric charts to determine a percentage of humic acid within that, right? So, and I see huge variability from sample to sample, and where I'm seeing the highest numbers is, of course, in compost. 3 or a 4 is really common for fields. But if you could be up at a 5, that'd be even better. The next thing I'm looking at when I'm beyond like counting and identifying the different nematodes and the flagellates and counting bacteria, one of the things I'm looking at is micro aggregates. So groupings of the bacteria with the organic matter and the fungi forming micro aggregates that I can see under the microscope. You can see these micro aggregates. So, and depending on the soil, you'll have lots of those, you won't have many at all. So your soil, because it's kind of this sandy soil that's leaching things and not a huge amount of...
There's no glue.
Yeah, there's not a whole lot holding it together. So your aggregation was not great. I wasn't seeing a lot of those micro aggregates under the microscope. And then here I have your micrograms of bacteria per gram of soil. So I have calculations and spreadsheets that I use to come up with these numbers based on the 5 fields. And the FB ratio, though it's important, and we're talking about succession, it's from my perspective, it's not anywhere near as important as having predators. Having your predators is the ticket to nutrient cycling. Having good nematodes, having your flagellates and your amoeba present in that system are critical to nutrient cycling. And the FB ratio, if you're in a system where you're encouraging succession, it will adapt. It'll change as you change your process. It'll improve as the fields improve. So when we're looking at these numbers, from a reductionist standpoint, from the way that most people like to look at data, we're looking at what is the ideal, what is perfect, right? I want to aim at that. What do I need to do to get there, right? And it's a flawed perspective, because understanding that none of these are 100% accurate and that there's variability and that it's dynamic and this is only a snapshot in time, these reports do not reflect 100% accurately what your field's like now. These reports were from a while ago, right? So, but they can create a baseline of understanding. So when I say baseline, that means where we started, right? And then when you make improvements and we do follow-up testing, now we have something to compare it to. We can see, yes, we did improve. It's much better than what it was before. I didn't have nematodes, now I do, right? I didn't have much fungi, now I do, right? And that is the why this baseline approach is important, so that we can create a system of continual improvement, and we have a way of tracking that.
So this is the reason why I'm doing this huge baseline report with Brian and taking all these soil tests, spending this time to do this, is because I'm really interested in showing more evidence of these regenerative and natural farming techniques working over time. And we just need more science. We need more evidence to prove that all this works, to help encourage other farmers to do this. And that's why we're putting this video out for everybody too, so that you guys can learn more, so that you guys can take your own soil tests and start compiling data that we can use to show that this does in fact work, and have data to also back it up. And I'm hoping maybe someday I could get some university to be interested in studying this, or help someone else get a university study done about these type of techniques, where we're combining all these different things together in a holistic view of our soil and our fields and forests and all of this. So, all right, let's look at the next report.
This report, the full baseline report, this is my aggregate report of the Logan reports, my physical testing that I did, my microbial testing that I did in my lab, and I combined them all into this report. So we've been reviewing all the reports, but generally for when I'm working with clients, this is the report to reference. If you don't understand any of these numbers, you want me to help you understand it, the full baseline report is the one that's been broken down to its simplest form and ease to understand. That's the whole point of this report. So we'll start right up here at the top. So fungi, there wasn't any. You want to aim for 1-to-1 in terms of an FB ratio. Bacterially dominated. It was mainly bacteria. Predators, there wasn't any there really, so we want to aim to get some of those. Was there anaerobic conditions? No, not really. Earthworms, there's NA here because I didn't take the samples. So if I would, generally I like to go out on site. We do other testing as well, and one of the tests we do is we'll take a spade of soil and we'll count how many earthworms are in there. And that's something that Stephen can do himself out on site.
I've been getting about 0-to-1, 1-to-2 in each shovelful, something like that.
1-to-2 is a good start. To give an example of where we could be, in one spade of soil, we could see up to 50 worms in one spade of soil in a really great system, right? Then we come into recommendations, and I start with microbial, right? So in terms of what I think this could use in terms of improvement, liquid IMO is going to be very helpful in improving this system, because when we add liquid IMO, and liquid IMO is made from IMO 4 generally, right? It contains not only fungal spores, some fungal hyphae, it's going to contain some of those predators, and if you made really great IMO 4, it should, right? And so applying that at 20 to 25 gallons an acre, 1 to 4 times a year, and usually doing this in summer or fall, right? And so when we're starting off the season, though, this is where we're coming into this differing perspective, right? So liquid IMO, though there is anaerobic fermentations involved in the process of making liquid IMO, it is an aerobic tea. It's compost tea for the natural farming world, right? But JMS, John Microbial Solution, is an anaerobic solution, and it's mainly facultative anaerobes, right? So these facultative anaerobes have the ability to prime the system, right? Really get that system going. And there's tremendous amount of yeast. I look at JMS all the time under a microscope. There's a lot more there than people think there is. There is fungi in there. There's other stuff going on in JMS. But the thing that's really great about JMS is its ability to break up compaction, its ability to trigger quorum sensing among all of the other bacteria and fungi that's there, and stimulate and jumpstart that system. So this is why JMS is recommended in the spring. Start off the year with JMS.
And that's those lacto and those yeasts are those things that are helping to break that up, right? Is that correct?
Yes, also other facultative anaerobes, ciliates and other, there is other, if you made a really great JMS, you should see predators in your JMS as well. And those guys are going to help to break up that compaction and prime the system, right? And likely, because of the context of your field, there more than likely is a compaction layer a foot or so down because of the practices that were used on your field before you were there, right? So JMS will be very helpful with that. And I'm recommending that once a year. You could go twice, but one time of a really good application of JMS can be very helpful, and then doing your follow-up microbial hits with something like a liquid IMO is going to be helpful as well. And both are about the same amount per acre, 20 to 25 gallons per acre.
So what I did this year, I did both of those applications. I did a stronger dilution of JMS. I just did the one that Master Cho recommends, and I didn't want to drive out 1,000 gallons of water. Like it's just too. It was too. I didn't have the right tank, and I'm still figuring out scaling this up. I timed it with the rain, and I let the rain do the dilution so that I didn't have to. Do you like that, or do you think it's better?
So they're Master Cho, and when they talk about JMS, get it down deep. Right? And so timing it with the rain is really great, because you have that low CEC soil that it will penetrate. It's sandy soil, right? So that is a really savvy way of doing it, of timing it with the rain to get it to saturate down deeper. Makes a ton of sense.
Okay.
And so those are kind of the main microbial things I'm recommending. Of course, you could benefit from putting out granular IMO or all like, but when we're talking at scale, that's a lot of IMO, and that's a lot of time and money. So generally, when we're talking at scale over an acre, we're going to be using liquid applications because it's far easier to scale that economically. So looking at chemical analysis, right? So all the things we talked about in those other reports are reflected here. And this is reflecting both the saturated paste and the Malic 3 report in this chemical analysis. It's on the full baseline. So I'm considering both tests in what I'm putting here. So in general, you're in a pretty good range with pH. Nothing to worry about there. I'm not an advocate of adjusting pH anyway. You're in a decent range for phosphorus, and I say this, though the saturated paste showed that you were low in phosphorus, I'm saying you're in a good range because I know that the biology is going to be able to mine those things. And then I put a little note here, low in field 3, a little bit of an outlier, but when we consider what we were talking about, that's the highest point, that makes a ton of sense, right? You're low in sulfur. So a sulfur supplement, something like a Jadam sulfur, highly diluted, could work with that. In a more traditional setting, something like Tiger 90 or other things could be used, right? But we're talking straight natural farming
wood
here.
ash a possible?
Yes, wood ash is going to be more of a potassium supplement than it would be a
That's true. It's high in
sulfur.
potassium.
So when we're talking all natural farming inputs, Jadam sulfur is often thought of as a pesticide, right? And that's the main way that the literature explains it. But if you do read the section on Jadam sulfur in the Orange Jadam book, it does mention that you can use this as a form of mineral nutrition for your plants. It's plant food as well. So, so that is what I'm recommending. And because you have low CEC soil, right? Because your soil doesn't have the clay content to hold on to a ton of nutrients. Our feeding techniques, that tells us a lot about how we would want to feed, right? So because you don't hold on to a lot and you leach easily, we kind of need to spoon feed a little bit until you start to get your organic matter higher through your cover cropping techniques and running the animals, and all the things you're going to do are going to raise that organic matter. And then over time, you won't have to spoon feed as much, right? So, and then I'm also considering the context of what you're doing with the field when I'm coming up with all these recommendations. So you're running animals on that field. So incorporating dry amendments, very challenging when you're running animals. So using liquids is a lot better, but also to coming back to the animals, when you have soil that's lacking in trace minerals, that's their food. The forage itself is lacking in trace minerals now, and then their nutrition is going to be lacking, and then it goes across the board all the way to us because you're going to end up eating the animal at some point. So when we're beyond, so in this spoon feeding scenario, we're seeking to amp up that nutrition of your forage, but in the meantime, also a trace mineral supplement for the animals makes sense, right?
Absolutely.
This is something that I learned once I started raising sheep that blew my mind over learning all this stuff. There's 3 methods of like remineralizing soil. The biology could do it. We could do Albrecht and just measure it and then add back in what's missing. The third way that I learned it with raising animals, grazing them, is the animals can put it back through supplementation of their diet. So you can feed them like what Greg Judy does. He has like a 26 mineral feeder. So 26 different minerals. The animal knows by nature which mineral it's lacking in. It goes in, it eats that, fixes the deficiency in its own body, poops out the excess onto the soil. So now as you're rotationally grazing these animals, the manure is leaving behind and the animal's actually balancing the minerals through this process. Now, that's a very expensive mineral system. What I do for my minerals, I do a sea salt and garlic, that's in one bin. Diatomaceous earth in one bin, and the third bin has organic kelp. And so the salt and the kelp are just kind of a broad spectrum. Diatomaceous earth is to help with parasites, and it has a few minerals in there as well. Now, another thing I thought about too, which is so interesting, another test that you could do is that doesn't require technology. Watch the plants that your animals are eating. Some of these plants are higher in magnesium in the leaves or higher in potassium or whatever. And you could kind of determine some more information about your field by observing what they're eating or what they're not eating. And the final thing we're going to look at is the plants growing. That's the other variable that tells us naturally what's happening in our soil.
Absolutely. So you're in a decent range with calcium, and I say that considering both the different tests and also the biology, because the biology is able to mine the calcium that you have.
Okay.
But because of that, because it's not in a hugely available form, I'm also recommending a little bit of WCA as a spoon feeding method.
That's the
While
spoon feed, yeah.
while you're amping up the soil, you're still spoon feeding a little bit until you get to that point where your fungi really is solubilizing a lot of that calcium. You'll still need to supplement a little bit. You're low in potassium. So for potassium, I'm recommending a JLF of Sulpomag JLF. So this is on, I believe, page 210 of the Jadam book. So this is essentially taking a mineral nutrition that people apply granularly, right? But we're using biology to solubilize that into solution and scale it up so that you now can use very little of this in a very strong and available form through biology, right? So going down the line here, you're a little low in magnesium. The Sulpomag JLF is going to help with that as well. Sodium, fine range. You have about 70 pounds an acre of nitrogen available through your organic matter. So there's calculations that we do from organic matter to convert that to estimated available nitrogen. The lab can do that for you. I can do those calculations myself as well. So in terms of nitrogen, you're in a decent place, but we could supplement with something like an FAA or a fish hydrosylate as needed kind of scenario. And then you're low across the board in trace minerals. You're in a good place for iron, very common. Your soil's kind of red, that tells you a lot. But in terms of trace minerals, fermented seawater is a good option. And that's generally something we'll do in the fall. And that's a one time a year. And I say one time a year because liquid IMO has sea salt in it. So you're getting trace minerals from your liquid IMO as well.
And JMS.
And from your JMS as well. So a one time a year of a fermented seawater kind of end of season, cheap kind of supplement to end that season as you come into the winter could be very helpful.
I had a question about, so if we inoculate all this fungi, these minerals in fall, now over the winter, how much does fungi slow down in cold temperatures? Because it is still able to move and facilitate things, but it's pulled back a bunch, isn't it?
Yeah, so it's still functioning, it's still there. The colder temperatures won't necessarily kill it. So what will happen is it'll go dormant and it will sporulate. And a lot of the same thing will happen with other biology as well. They'll kind of go dormant and sporulate. And that's why that spring hit of JMS to wake everyone back up is really helpful, right?
And maybe an SOS even, the soil treatment solution. That's like in Korean natural farming, that input's designed for what he's saying. So that combo would be really good.
Oh, absolutely. And ending the year with something a little kind of simpler, like a fermented seawater, could make sense just in terms of economics of not having to use all those inputs for when you're closing out the season a little bit.
Makes sense.
And then the sulfur, the Jadam sulfur. So highly diluted, 1.5 cups of JS to 100 gallons of water. Very, very diluted, 20 gallons per acre, 2 times a year. So spring and fall. So close out the year with it, start the year with it, and get a little bit of your sulfur supplement in there, right? And then the Sulpo Mag JLF. So this takes 3 months to make. So you're going to want to make that in the winter time. And as it gets older, it's going to become a little bit stronger and you'll be able to dilute it more. So the dilution rate, this is based on what you're seeing in the field. So this is based on the nutritive cycle, what your plants are telling you. So it does require listening to your plants to come up with the dilution rate for this, but it could be anywhere from 1 to 50 to 1 to 500. And for JLFs, I highly recommend using an EC meter to track the strength of that JLF to make sure that you are diluting it enough. So that'll be variable depending on, did you use leaf mold or did you use IMO4 to make that JLF? How old is it? All these different things. Did you use rainwater? Did you use other water? So there's variability in what the JLF will be and how strong it'll be. So, but this is a JLF that's literally like you're buying a bag of mineral nutrition, but you're using biology to process that and to make it in a super available form that's now very economical because it doesn't require very much of it at all.
And that the EC meter that he mentioned, that's like I think it's a $100 device, and I'll put a link in the description if you guys want to check that out.
So the one that I like for this application is the Blue Lab truncheon meter. Is very common. Microbes by Marco advocates that. A lot of people are using that Blue Lab truncheon meter for this context.
For hydro too, right? Is good for that.
Oh, absolutely. And what it's doing is it's showing the amount of available salts. So cation and anion attached to each other, those are available nutrients. So JLF is a highly available nutrient, but it's because it's been processed by biology that's making it so available. And because of that, it's used very diluted. It's incredibly strong. This stuff can kill your plants just as easily as it can make them better. So using JLFs with a lot of understanding and diluting them properly is really important. But it's a very economical way of scaling up very available nutrition. WCAP, as needed, right? Matched with your nutrient cycle. And I would recommend, if you need a little bump on something like that, and that's based on what you're seeing in the field, you're going to add that stuff to your liquid IMO application. I wouldn't do it by itself. You'll add that in. Same thing with WCA. You need a little bit extra calcium because you've got stuff in flower out in the field, or you've got things that are where you're seeing a lack of nutrition. So what'll happen is when you're lacking in calcium, you're going to be lacking in everything else too. So when you hit it with a calcium, it makes a lot of sense to maybe that next day or a couple days after, hit it with a trace mineral, right? So because what you're doing is you're opening up the calcium pathway to allow all of those trace minerals to become more available now. So when you're doing a liquid IMO, you're kind of doing them at the same time. If you add calcium to your liquid IMO, you're opening up the calcium pathway, but you also have the trace minerals from the sea salt in your liquid IMO. So you're kind of doing both. The pathway that I'm talking about is in the cellular structure of the plant. So you're opening up the calcium pathways within your plant, not necessarily within the soil, right?
Got it.
And so these are all as needed, matched with the nutritive cycle, right? So matched with what you're seeing in the field. So if you're seeing like your plants aren't very green and you have a nitrogen deficiency, adding a little FAA into your liquid IMO could be helpful. But these are as needed kind of things. The other things I'm recommending are highly recommended, where these other ones are if you need them kind of scenario. And one thing to keep in mind with FAA, so A lot of people who make compost tea like to put fish in the tea while they're brewing it. There's not a lot of science to prove that that's actually beneficial. And when you microscope it, you'll actually I see lower fungal counts when I do that. And what you'll see when you put fish with a bubbler in there is it's going to bubble up like crazy.
Yeah.
And that's a misconception itself, that bubbles equal biology. A lot of the time the bubbles are from sugar. You put a bunch of molasses or you put a bunch of fish, your thing will bubble over. I guarantee it, even if you didn't put compost in there. But so when we're talking about adding FAA to a liquid IMO, make your liquid IMO and then add the FAA at the very end. Don't brew it with it.
Right.
So
Okay.
those are things the just little tidbits that are helpful. But
Okay, that's a good tip.
the last thing that I'm looking at is at the bottom here, physical properties. So this is sandy loam. So using simple jar test, you're about 55% clay, about 55% sand, about 35% silt, and about 10% clay. That makes sense, right? When you look at that CEC, there's not much clay there.
Right.
Humic acids, you're a 3 out of 6. Organic matter, you're below 5. So being below 5, not the best, but we want to aim for being at about 4 or higher could be great, but if we get at 4, that's a pretty good range that a lot of certified regenerative farms and things are at about a 4 to 6. Seeing higher numbers than that, you may see a really high number if you're in a scenario where you're adding tremendous amounts of compost every year. Sure, you could see higher numbers, but in a pasture setting or in a field setting where you're not gonna, who's gonna add all that compost? That's a lot of money.
Right. Yeah, you're
So,
gonna see those higher numbers in a garden system. That's where you can see those.
Totally. And then in terms of infiltration rate, so I can take these percentages of sand, silt, clay, I plug these into charts and I can create an estimate of your infiltration rate. So you're at about 1 to 2 inches an hour-ish,
It's not good. I know it's not good from observing the rain.
Right. And there's a reason for that. You would think, so sand has the ability to leach a ton of nutrients, but sand can hold a lot of water, right? So if you've taken a bucket of sand, fill a bucket up with sand. Leighton Morrison talks about this, and start filling it up with water and see how much water that sand will absorb. It's a lot. It's pretty impressive. So that has a lot to do with this infiltration rate. Because you have high sand, low clay is leaching the nutrients, but the sand is actually holding on to a lot of water, and it's not necessarily going to penetrate the way that you would think it does. If you've walked on the beach and you can feel sand that's fully saturated, so it's kind of sticky and your feet sink into it. But so this is the full baseline report with recommendations from all the other testing that we did, and is the main report that I look at with clients to help them understand what it is that I think they should do. And when it comes down to it, your perspective of what you should do on your farm is your decision, right? And what I encourage is that people understand that they are part of the farm and that their decisions mean a whole lot. And so when I'm giving these recommendations, these are based on estimates, and generally I've been to Stephen's property, but we didn't do these tests. We didn't collect these samples together, right? And generally what I'd like to do when I'm working with a client is we actually get out there in the field and we start feeling the soil and we smell it and we touch it. And these are all things that Stephen does himself. But when I'm tailoring these recommendations, I'm considering these other things that don't have anything to do with lab testing. They have everything to do with what I'm seeing in the field. So an old adage from Gary Zimmer, a world-renowned soil consultant, is if the soil test is telling you one thing and the field is telling you something different, throw the soil test away, right? And I highly encourage everyone to use critical thinking and to look at what's happening in their soil, be in tune with their plants, be in tune with that nutritive cycle, and you should never apply anything blindly onto your field because someone else told you to, even me, right? You should do it with understanding and why you did it. And I'm here to help people understand why, from my perspective and my experience. But at the end of the day, you're the master of your field, and the best fertilizer is the footsteps of the farmer. It's you out there paying attention and getting in sync with what's happening with your plants and your animals and your field and your environment. One of the other things that I like to do when we're looking at a field is look at the weeds that are growing in the field. And weeds can tell us a lot about what's going on with the field as well. And this is based on research that other people have done around the world who have collected different weeds from the field, sent them off for tissue samples, and then aggregated data to show what, in general, different plants indicate about what's going on with your soil over a number of parameters, right? So in general, Bermuda grass, which is one of the things that's growing in your field, shows low calcium. Also, low phosphorus, low magnesium. And what you notice here is there's things that are blank. So the data is not necessarily available for every single nutrient for every single plant. And the data is not available for every single plant either. So Steve Reisner from Potent Ponics, he's starting to create a database of different plants and the nutrition that they have in them for making FPJs, making JLFs out of them. So there's research being done, but it's not necessarily available for everything.
I know there's in permaculture, there's the dynamic accumulator list. I don't know who put that together, but I've seen that before, and that has a good list of nutrients as well.
I think that's from Duke University or something like that. But the Potent Ponics he's created a database that's quite extensive. It's one of the most extensive that I've seen. And these are things, when I'm looking at this, does this line up with what the other testing is saying? And it doesn't always, but it's interesting to look at because this is saying, it's indicating that Bermuda is low calcium, right? Bermuda also breaks up compaction. So this could say that what I speculate is that there is a compaction zone that's lower down in your field, right? And Bermuda helps to break up compaction. So when we're looking at weeds, we're asking, why is this weed here in succession? What is it trying to do to help the land? Because all weeds are trying to help the land and have their place in succession with what will come next, right? And so Bermuda grass in general is an indicator of low calcium, and low calcium can be an indicator of compaction, right? And so that's something interesting that we're looking at when we're looking at Bermuda. Also, it's an indicator of low magnesium, low phosphorus. So our malic 3 is saying we're high phosphorus, saturated paste saying we're low, but what the plant, and that lines up actually, because what the plant is getting is what's available, right? So, so this is an indication of low available phosphorus, right? It's an indication of low available calcium, right? And our testing is showing that as well. So fleabane, same thing, indicating very low calcium, very low phosphorus. Interestingly, this is an indication of high potassium and high magnesium, which is different than what other plants are telling us. So this is not always going to line up perfectly.
And if we were to take a tissue sample of my fleabane and send that off, we might actually see that it's low in magnesium because my field isn't able to give it the magnesium or can't get access to the magnesium.
Doing tissue testing on the forage in the field is a very interesting thing to do, and that's really is the next level of what we would be looking at. And then you're in a scenario with your pastures where you have you're in an area that has rain, So you're not necessarily watering your pastures regularly. But another thing to look at is the water test, because the water that's going onto the field has a lot to do with chemical nutrition as well. So ironweed, again, low calcium, low phosphorus, indicators of low humus, every one of them. So that lines up with the fact that we have low organic matter in this field, right? And all of them are indicators of low calcium. Not necessarily all of them are indicators of low phosphorus. Not all of them are indicators of high potassium and high magnesium. That's an interesting thing with fleabane, and would be one that would be interesting to do tissue testing on because of that. And also these fleabane and Johnson grass are indicators of high iron.
That's very common in this area of the country too.
Absolutely. It's very common for high iron in that area. So this is not every single weed, because some of the weeds that Steven sent me to do research on are not ones that I have information available to me about. So this is an area where there's tremendous work to be done. There's much research to be done in this area, and participating in one of these databases we talked about, that's an open source database where we can do some of this tissue testing to make this information more available to everyone is an interesting thing to do. So looking at these weeds and what they indicate tells us a lot. It's another window, it's another snapshot to widen our view of what's happening in the field. Other plants, just to give an examples of some of the things that they can indicate. So say you have milk thistle or thorny plants. We'll use milk thistle as an example. Milk thistle, if you study holistic health, you know that milk thistle is something that's detoxifying for your body, right? And these plants that are detoxifying for our body are also detoxifying to the land. So if you have a ton of different thistles and thorny plants, generally these nastier plants are the most detoxifying growing on your land. That can tell you a lot about contamination on your land. So I've looked at a lot of different properties where I've seen milk thistles or different thorny plants growing in a certain part of the yard, and then I'll ask the person who owns the land, is that where your septic leach field is? Or did you dump something there? What's the history of that land? And more often than not, it is where their leach field is, or it is where they dump something. So that's the land's way of detoxifying that waste, right? And so there's really interesting things beyond just these minerals and this chemistry that the plants are trying to tell us. And if we're able to listen, again, integrating ourselves with this, allowing ourselves to have the ears and eyes to see from a different perspective what might be going on with our land, we can gain tremendous insights that we can then create actions from.
I love that. I love that. I was going to add one more thing to this. Those weeds that or the plants that we just talked about, those are all plants that my sheep, that's not the first thing that they go for. They go for that later on after they've eaten their other favorite things. And then the theory for that would be, well, because these have less nutrition in them, or less of the things they're looking for.
Absolutely. And you can do other interesting things, like doing a Brix reading on your weeds, and then doing a Brix reading on the forage that they like, and comparing them could be really interesting to see what is. So Brix don't tell us specifically what's there, but they tell us total dissolved solids of sugars and mineral nutrition. And so we'd like to see something higher than 12 in terms of Brix, right? So if we're seeing really high Brix in the things that they like and lower Brix in the things that they don't, that's an interesting metric to consider when we're talking about animal forage and animal nutrition. Right? The Brix is cool for on-farm because tissue testing can be quite expensive, and Brix testing is free if you have a refractometer. So those are all of the tests that we did. So if we were to do more testing, right, I would say water testing would be interesting also, but it's not as important with your context because you're in an area that gets rain and you're not actively watering your pastures, right? And then also tissue testing is kind of the next level of seeing how all of these things are now affecting the plant growth. So this is what's going on with the soil. These are things that I did, right? These are actions I took. How did this affect the end result of what's in the forage now? And then also drawing transects on the field of, in this area, since I've done this, this weed is starting to go away and this one is now growing more, you know, and now the crops that my animals like the most are more abundant than the ones that they like less, you know, and so drawing out areas and then taking a 2 foot by 2 foot square of that area and pulling it up from the roots and seeing how much root matter is there. How tall did it get? How much biomass was available in a 2 foot by 2 foot square of this field? And how does that compare to the rest of the field, right? So these are other things that we can do to widen our view of our understanding what's happening. And then even beyond that, when you get to the point where you harvest your animals, you could send off testing of the meat and see the nutrition that's available in the meat itself.
Yeah, that's where I'm trying to go. And
This is why I want to make more money, because I could like pay for all, you know, this is thousands of dollars a year to do all this stuff. So it ain't cheap, but this is like, this is where I'm going for myself, for my content and my future is showing all this stuff. Because like, I already know that there's way more nutrition in the eggs that I raise, the meat that I raise. Like, I don't need science to tell me this. It's obvious, but I'm just so interested in convincing more people, more farmers that this is the most legit way to farm. And yeah, I think it's very cool. So thank you for doing this with me.
Absolutely. With all the testing we talked about is like, though there's a cost to it, it's helping to build a true holistic context, right? And if we were to go even further than that, right? So you did all of your testing on your soil, then the plant material, then the animal itself, and then you consumed it. You could do blood testing on yourself, right? There's even another thing you could do, you know, to see how it's affecting your health. And then we take this holistic approach on ourselves, and then also tracking other aspects of the soil food web. So since I started to do this, I've seen more animals, I've seen more insects, I've seen plants coming up that I haven't seen before. You know, things like that are other things we can do to widen our holistic context and gain a deeper understanding of what's going on.
So a journal, little mini journal, I guess, on my field and what I'm seeing and what's going on. And okay, I'll have to start doing that too. That was a super in-depth look of what it takes to understand a soil test and all the different variables involved. So thank you so much, Brian, for doing this with me, educating us. And I know that everyone watching this has taken away multiple things that they can go research more or apply to their own soil. So if people want to get in touch with you, if they want to recruit your services to have this exact thing done for them on their land, how can they get in touch with you? So
the best place to find me and to reach out is via my website at earthgrowerllc.com. If you visit the website, all of the links to all of my other social medias are there: Instagram, Twitter, YouTube, Discord links. But my Instagram is earth_grower. You can see some of the projects that we're working on there. But the best place to reach me would be at my website. And if you go to the Instagram, you're going to find the website. If you go to the website, you're going to find everything else. So
there'll be a link. I'll put a link down there too.
Yeah. If you go to my website, I have a ton of information on there as well. I try to make it kind of a rabbit hole of information. If you're looking for even deeper content on some of the things we're talking about, on my Discord page, I've kind of aggregated videos and stuff from all kinds of people. Some of Steven's videos are on there. There's tons of videos from everyone else, where I've kind of compiled them in one place, where people can find them.
Cool. Thanks, Brian. Well, thanks for all of the great work that you're doing in this space, and just keep it up. And definitely look forward to some future videos on my channel with Brian. I hope to bring him back on next year when I do the next part of the soil test. I'm going to do his recommendations, and then we'll see what happened as a result of all that. So I'm really excited for that update.
Absolutely. Yeah, thanks for having me. I appreciate it, and I'm excited to see where it all goes.